Yes, we're good. Okay.
Yes.
Sharing. Do you see me scrolling? Everything's good. Excellent. Yes, so thank you, Dennis, very much. I didn't expect such an extensive introduction, and I really appreciate it. It was really a pleasure working with you and at the University of Colorado. That's why I'm still here. So also I would like to thank, obviously, the organizing committee and the leadership of the Gates Institute for inviting me to present our work. In particular, and I'm sorry if I don't list everyone, it's Jill, Debbie, Jane, Laura, Diane, Terry, Dennis, and the entire advisory board, since we wouldn't have been where we are without your support, without Grabstake Awards, and without provided infrastructure. So today I will present the work that has been done in collaboration with Drs. Igor Kogut, Denis Rub, and Anna Bruckner. As you could see from the title of my talk, we're developing a stem cell therapy for inherited skin blistering diseases. So before I continue, let me again mention a few potential conflicts of interests. So as Dennis mentioned, our RNA reprogramming technology has been patented, and our patent has been issued on October 4th, 2022. Another technology, our combined gene editing and reprogramming technology, has also been patented, and the patent was issued on January 31st of this year, very recently. We have non-exclusive license agreement with Selena Biotech to adapt our combined gene editing and reprogramming to their AI-based platform. We also completed a sponsored research agreement with a potential licensing option with Avita Medical. So since the Gates Institute is interested in developing stem cell-based therapies for a variety of diseases, let's discuss the limitation for using adult stem cells in the clinic. So we know that adult stem cells have limited proliferative capacity. Basically, it's very hard to expand them in the dish, which creates difficulties in scaling up for manufacturing. Also, there is limited availability of different cell types for transplantation. Think about neurons or cardiomyocytes. These cells are not easy to get from the patient, and obviously we cannot even expand them in the dish, so that's a problem. Also, there are ethical concerns if embryonic stem cells are used instead of adult stem cells. However, the majority of this limitation has been recently addressed by the development of induced pluripotent stem cells, or iPS cells. So iPS cells are generated by transient delivery of just 4 transcription factors, OCT4, SOX2, CALF4, and c-MYC, into somatic cells. And this is sufficient to reprogram these adult somatic cells back into embryonic stem cell-like state, which we called induced pluripotent stem cells. Because iPS cells are very similar to embryonic stem cells, they can be expanded indefinitely. They are amenable to genetic correction. They can be differentiated into any cell types in the body. Again, thinking back about neurons and cardiomyocytes, that's the source of cells of cardiomyocytes and neurons. These cells are also patient-specific, so that should decrease the risk of immune rejection if we ever transplant iPS cell-derived cells back into the same patient. And obviously, there is relatively low ethical concerns associated with the use of these cells. So how can we use iPS cells and research in the clinic? Let's assume you have a patient with a disease, which you would like either study or develop a therapy. So we can easily make induced pluripotent stem cells from this patient. For that, we can collect either skin biopsy or blood specimens or urine and reprogram cells derived from these specimens into iPS cells. Because iPS cells can be expanded indefinitely, you in a way create a renewable source of patient-specific cells. So then, if you differentiate these iPS cells into target tissues, again, think about skin, neurons, cardiomyocytes, hepatocytes, then you can create a disease modeling system. So in a way, you can study the disease mechanisms. You can also use these iPS cell-derived cells for in vitro modeling or in vivo modeling, where you can test your novel therapeutics to treat the disease of interest. And eventually, these novel therapeutic strategies can be applied to the patient. However, if the disease you're interested in is caused by a known mutation, what you can do, you can perform gene editing on these patient-specific iPS cells, generating genetically corrected iPS cells, which are still will be patient-specific, so they still will be autologous. Then you can use these genetically corrected iPS cells, differentiate them into target tissue, and transplant back to the patient as a therapy. So obviously, iPS cells are still relatively novel technology, and it's new for the FDA. So FDA is very cautious about using and approving this technology for the treatment of the disease. So therefore, we really need to pick the right model to test the applicability of iPS cells for therapies. And in this regards, skin may be an ideal tissue to try and basically apply an iPS cell-based gene correction therapy. So the skin is readily accessible. It's easy to monitor. So if after transplantation we see tumor being developed, we can easily excise it. It's feasible to transplant. We know how to transplant skin for decades now. And the most devastating and incurable forms of inherited skin blistering diseases are monogenic disorders with no causative mutations. So we can correct these mutations on the DNA level. So looking at the skin, we decided to focus on recessive dystrophic epidermolysis bullosa, or RDEB, as our first model. So RDEB is caused by the mutations in collagen 7, which is the main component of anchoring fibrils that connect epidermis and dermis together. So this cartoon shows you the structure of the skin. So primarily it contains two major layers, the skin. It's the epidermis and the dermis. Epidermis also is a multi-layer structure. The basal layer of the epidermis contains keratinocyte stem cells. And keratinocyte stem cells, they differentiate and they go upwards where they create the suprabasal layers. And eventually we get stratum corneum. That's basically what you feel when you touch your skin. So each layer of the epidermis expresses its own markers like basal layer K5, K14, suprabasal K10, K1, and loricrene like the top layer of the skin. And the reason I'm describing these markers is because later in my talk, you'll see we use them as the markers of different layers of the skins once we basically make a new skin from iPS cells. Does anyone has question? No? Okay. And then basically, then we have a basement membrane, which serves as a glue, which connects epidermis and dermis together. And that's where collagen 7 plays an important role. So you can imagine in these patients, because there is no collagen 7, there is no functional basement membrane. So the epidermis can easily get separated from the dermis. And the patients develop blisters, which look like that. Once these blisters heal, they basically form scars. And eventually, as the patient ages, the entire skin looks like a scar tissue, as you can appreciate from this image. And it's all because of the lack of collagen 7. So here I'm just showing you examples of skin biopsies. On the left is a healthy skin from the healthy patient stained with collagen 7 antibody. And you could see this is where collagen 7 is. This is the basement membrane. This is epidermal layer. This is the dermis. In RDEB patients, we don't see any collagen 7. So in addition, not only you can imagine that these patients go through lots of pain, they are subjected to inflammation, they are subjected to infection and eventually sepsis. These patients also succumb to very aggressive skin cancer in their early 20s, and that's their main cause of death. So you see, this is very devastating disease, which we decided to use as a primary target of our iPS cell therapy. So also, again, this is a recessive disease, and usually all our genes have like 2 copies in the genome. So both copies of collagen 7 gene in these patients are basically mutated. However, we know that parents of these patients are not affected. So for our therapy to be effective, we technically need to correct only one allele in these patients. So importantly, we need to select the right cohort of RDEB patients for our future clinical trial. And it's very important because collagen 7 is like RDEB is caused by mutations, like multiple mutations in collagen 7. So like all over the gene. So, but University of Colorado has been involved in EB care and research for decades. We collaborate with Dr. Anna Brockner, who is the director of EB clinic at Children's Hospital. She has access or sees at least 39 patients with RDEB. And 6 of these patients share the same founder mutation in intron 98. And it's very important. You see, we now have access to the patient with the same founder mutation. For the FDA may consider each gene editing strategy as a separate drug. So basically developing a strategy for one founder mutation kind of opens the door that we can apply the technology to other diseases. So what this mutation does. So as I said, the mutation is in intron 98. So it affects splicing. When we did RNA sequencing of our patient specimens, we find out that the majority of transcripts in these patients have intron 98 retained in their transcript. Here you have a premature stop codon, so obviously full-length collagen 7 is not produced and it's being degraded very rapidly. However, very little number of transcripts, less than like around 1%, retain 15 nucleotides from intron 98. And these 15 nucleotides be translated into 5 amino acids in frame with the rest of the collagen 7 protein. So obviously this read-through transcript is not functional because this additional 5 amino acid affect very important domain of the collagen 7 protein. However, it means that immune system of these patients saw the full-length collagen 7, at least at one point or another. So again, this is a recessive disease. This patient immune system never saw collagen 7. And now all of a sudden we reintroduce the wild-type variant of collagen 7. So there may be a risk of rejection because of this reintroduced collagen 7. But again, because these patients basically express this full-length protein or in transcript, then we hope that this will diminish the risk of immune rejection because of the reintroduction of collagen 7 into these patients, making this cohort of patients well suited for our gene correction therapy. So let's look at the schematic of the whole iPS cell-based technology, iPS-based therapy. So again, we have a patient with RDEB. We can collect skin cells from this patient, reprogram these cells into iPS cells. Then we can perform genetic correction of these iPS cells, generating and select these genetically corrected iPS cell clones. Then we take these genetically corrected iPS cells and differentiate them into keratinocytes and fibroblasts in order to reconstitute skin of these patients. So let's discuss each step of this therapy individually, starting with the reprogramming. So what would be the requirements for a clinically relevant reprogramming method? It has to be genome integration free. It should give us high efficiency to preclude selection of mutations. So we basically should have enough iPS cell clones at the end of the reprogramming to select the good one for future clinical applications. However, as I mentioned here, the majority of clinically relevant methods have the efficiency below 1%, which is very little. So it's not sufficient for successful clinical trial. The reprogramming method should produce stable iPS cell lines, and ideally it should start with small starting number of somatic cells. So we do not expand our somatic cells too much before reprogramming, not to introduce extra mutations. So in order to kind of achieve the goal and develop clinically relevant method, we actually improve and generate a new method, RNA-based reprogramming method. So we published this method in Nature Communication in 2018. And as I mentioned, and as Dr. Roop mentioned, we just had the patent issued for this technology. So this technology is non-integrating. It requires fewer cell numbers to initiate the procedure. For RDEB cells, we can use 3,000 cells. For healthy controls, we actually can reprogram a single cell. And it's very high efficiency. In order to make it to work, we created a cocktail of factors consisting of modified mRNAs, encoding modified OCT4, SOX2, CALF4, CMYC, Lin28, and NANAC. And I have to note that these modified mRNAs are kind of the same molecules or similar molecules that we now use in vaccines against COVID-19. Also, our cocktail contains mature microRNA mimics, 367 and 302s. We also optimize plating conditions and culturing conditions for our cells. We introduce these cocktails every other day for the duration of 2 weeks. It could be shorter for the successful reprogramming. And at the end, we detect iPS cell colonies. In the particular example that I'll show you in a second, we use TRA160 staining, which is pluripotency markers, so we can easily visualize all of the derived colonies. So in this example, we plated healthy neonatal fibroblasts at different densities in a well over 6-well format dish. So we plated 10,000 cells, 1,000 cells, 500 cells, and 200 cells. And you could see that the efficiency, we can get up to 800%. So we get almost 2,000 colonies out of 200 cells. This is possible because, again, we plate few cells, but these cells continue to divide and form multiple sister colonies. That's what we're dealing here. But anyway, the efficiency is extremely high. This is how TRA160 staining looks like under magnification. We also use this approach to reprogram, like as I said, this is healthy cells, but we tried it on adult disease cells. And the efficiency, of course, now is not 800%, which is expected, but it's still double digits, giving us sufficient material for clinical application and for selecting good iPS cell clones for potential application in patients. To validate that we're indeed dealing with pluripotent stem cells, we induce teratomas. It's just a simple injection of undifferentiated iPS cells into the mouse. And if the cells are pluripotent, they form tumors consisting of different cell types of 3 germ layers. So here I'm just showing an example how these teratomas look. So this is OCT4 staining of the colony. So this is just an iPS cell colony consistent of multiple cells clumped together. Upon injection, we get tumor with different tissues formed. Here is ectoderm, neuronal rosettes. This is mesoderm. This is chondrocytes, basically cartilage. And endoderm is just a simple epithelia from lungs or intestine. So we applied this protocol to our RDEB patients. So I'm showing an example from 2 RDEB patients. Both of these patients have homozygous mutation of this so-called Colorado mutation, the mutation in intron 98. But we have more patients which have heterozygous mutations. But anyway, we successfully generated iPS cells from these patients. You could see that the cells stain positive for pluripotency markers, non-OCT, OCT4, TRA181. We also karyotype these cells and confirm that they have normal karyotype. So there are no additional genetic abnormalities in these cells, which confirms that these cells are of good quality. So let's go back to our schematic of iPS cell therapy. So we discussed the reprogramming. You could see that there are extra steps here before we even can initiate gene correction. It's like a quality control tests. We really need to see that iPS cells are of good quality, express all the right markers, and only then we can do genetic correction. However, once we do genetic correction, we again have to repeat the same quality control tests. So we were thinking, can we try to combine reprogramming and gene editing into one-step procedure like that, cutting all of these extra steps and making the whole procedure is more straightforward and cost-effective. And that's what we try to do. And here we know that our protocol can be initiated with very few cells. So if we try to achieve genetic correction in those starting fibroblasts and then complete reprogramming process, the resulting iPS cell clones should be monoclonal. So then we don't need to do extra purification. We have already corrected monoclonal line derived from that original fibroblast. In order to achieve gene correction, we use Cas9. So I will not go into too many details how Cas9 works. You probably all know it. It introduces specific DNA double-stranded break in a specific locus in the DNA. And as long as we provide the correcting template with the healthy variant of our gene, we can basically have corrected collagen 7 in our patients. So in order to make the combined gene editing and reprogramming, what we did, we basically plated again very few fibroblasts, like about 3,000 cells. We introduced Cas9, assessed DNA, and guide RNA, and assessed DNA is our single strand DNA donor carrying the healthy variant of the sequence. We introduce it into these fibroblasts at the beginning of the reprogramming. Then in the same dish, without splitting the cells or doing any manipulations, we continue the reprogramming process. And within 2 weeks, we have multiple colonies that we can collect individually and analyze. And as expected, most of these colonies were monoclonal, so they did derive from that original fibroblasts that were plated at the beginning of the process. Here, I'm just showing an example of few, like just Sanger sequencing, showing that, for example, this colony had one allele corrected, while the other allele basically was unaffected. And it's totally fine, as I said, because we're dealing with the recessive disease and parents of our patients do not show the phenotype. We only need to correct one allele. And this is just clone that was not modified. So again, collected from the same dish. So our protocol is footprint-free. So we use Cas9 delivered as modified mRNA. We don't use any selection marker, and we use single-strand DNA donor to reduce off-target integration. So if we kind of collect that whole plate and see what is the overall efficiency of correction in our reprogrammed wells, it gives us 5% of efficiency per allele. So which is pretty high and allows us to only pick maybe under 100 clones, and we should have enough corrected clones to go ahead with clinical applications. So as I said, we technically, and this technology was already also patented. We just got the patent at the end of January this year. So technically, we now know how to reprogram cells and how to combine reprogramming and gene editing in a one-step procedure. So the next step is the differentiation of genetically corrected iPS cells into keratinocytes and fibroblasts. And this step, we really kind of had lots of issues. We spent probably most of the time trying to optimize this procedure. And here I would like to thank Marina Pavlova as a research associate in the lab, who really spent years trying to make it work. So, and basically, that was really a big hurdle for us. But now we think we succeeded. So the major hurdle we've been facing is inconsistent differentiation of iPS cells into keratinocytes and dermal fibroblasts using a monolayer culture. So we would just plate iPS cells, apply all the right cytokines. At the end, we would get cells which look identical to keratinocytes and fibroblasts. They would express the right markers. Everything looks great until we try to graft these cells and recapitulate, basically make human skin in vivo. And that's when they would fail. So technically, then we thought that normally during developmental process, our differentiation and development of our tissues happened in 3D environment. It's not just on a flat cell culture dish. So it's very important that the cells have the right orientation, they integrate with each other and basically get the right cues to differentiate. So that's why we decided, what if we try to differentiate iPS cells into keratinocytes and fibroblasts in 3D conditions? And luckily for us, in 2020, the paper came out reporting the generation of hair-bearing skin organoids. So technically, the researchers were able to find the right conditions to grow skin organoids out of iPS cells. Of course, it's a long procedure, it takes about 3 months to accomplish, requires lots of manipulation. But at the end, you get organoids that consist of 2 parts, the head That's where your epidermis and dermis is. And you could see that the epidermis face inwards. This teal color is where your epidermis is. Whatever surrounds it is the dermis. Importantly, these organoids also show the development of hair. So, and it's very critical. So there are hair buds forming in these organoids because we know that the formation of hair is the most stringent test for functionality. So if keratinocytes and fibroblasts can support the development of hair, we know that the cells must be functional. The other part of this organoid called tail is just a cartilage, which supports the head of the organoid. So in order to see if we can adapt this system in the lab and develop functional skin organoid, we generated a specific iPSC line by knocking in M-wasabi, or technically it's like GFP, fluorescent reporter, at the end of the keratin 5 gene. And keratin 5 is expressed in keratinocytes, but it's not expressed in iPS cells. So we know that when we successfully differentiate iPS cells into keratinocyte lineage, we should start seeing keratin 5 expression and fluorescence from this keratin 5. So it's like very nice lineage tracing model to detect that everything is going the right way. So as you could see, iPS cells, no green expression, no fluorescence. Once we subjected these cells to keratinocyte differentiation using our monolayer culture, the one that still doesn't give us fully functional keratinocytes, you could see that island start fluorescing green. It's around here, indicating that that's where we start seeing the commitment toward keratinocytes. So by doing this experiment, we confirmed that we have the right lineage tracing model. Then we use this iPS cell model, iPS cell line, and subjected it to the differentiation into skin organoid. So again, this work was done by multiple people in their lab, Marina, Sinead, Sean, Jocelyn. So they all work hard to make it work, but that's how it looks now. So this is the skin organoid. This is the head, basically this part. All of these weird things you see is actually developing hair follicles. If we check it later, like about a month later, we now see the actual hair growing inside the organoid. With a higher magnification, you definitely see kind of the forming hair inside the organoid. So it did work. We know that these cells should fluoresce green because, I mean, the epidermis here contains keratinocytes, and we should start seeing green color coming from keratin 5 expression. That's exactly what we see at the latest stage organoids, the one that have hair formed. You could see that they fluoresce green because that's where keratin 5 is expressed. So we know the system works. We basically collected organoids at different time points during differentiation, and that's how it looks. The green color, again, marks K5, keratin 5, marks your epidermal stem cells or epidermal cells. And you see this is the head of the organoid, so it fluoresces green. This is the tail, it's cartilage, and it's not showing green because there is no keratin 5 expression. So as we check the organoid later in development, now we see the formation of hair follicles. It looks a little bit different because now you see kind of the cells concentrated into the formation of hair follicles, again, suggesting that now we're dealing with functional organoids. So if we look at H&E staining, just histology of the organoid, this is the cartilage, very typical cartilage here. And this is your epidermis. It's still day 62, so it's a little bit early. Usually we now collect organoids day 85 and later. So we see the epidermis, developing epidermis. This is dermis, and these are developing hair follicles. So you can imagine your epidermis kind of facing inwards, not outside, but inwards. That's usually how most organoids develop. They all kind of face inwards, probably because of the way the cytokines are provided to these organoids. But importantly, we can dissect these organoids and isolate fibroblasts and keratinocytes. So here I'm just showing again the same K5 and wasabi iPS cell line that we differentiated, and then tried to isolate keratinocytes from, and you could see that it worked successfully. We see these green islands, which is our keratinocytes, and morphologically it looks like keratinocytes. So we were very happy with the outcome of this protocol. So eventually we used our genetically corrected RDEB iPS cells that were generated using our combined gene editing and reprogramming, and we have at least 3 different iPS cell lines from 3 different patients with the same Colorado mutation that we subjected to differentiation. So here again, I'm just showing you control. It's just a healthy iPS cell line that we, sorry, too early. Healthy iPS cell line that we differentiated into organoid. It's a later time point. You now see keratin 5 expression, which is red. And if you remember that cartoon I showed you at the beginning of my presentation, that's where knowing different markers of different layers is important. So keratin 14 is a marker of the basal layer of the epidermis. That's where we expect to detect keratinocyte stem cells. As they differentiate, they start expressing K10, K1. And here I'm just showing K10 staining. And you see it basically sits on top of K14 layer, which is exactly how we expect it to be. And then a little bit later marker of differentiation is loricrene. And you see it's being expressed here, suggesting that our keratinocytes go through the full commitment and are functional. Vimentin here stains the dermal component of the organoid. These are hair follicles here and the dermis. And this is collagen 7. Remember that's collagen 7, which is very critical for our RDEB patients. So we can easily detect it in our organoid, confirming that the basement membrane indeed is formed in these tissues. So then we differentiated iPS cells, uncorrected iPS cells that we derive from RDEB patient. So basically to see what is the baseline we're dealing with. So again, RDEB iPS cells formed nice organoids, they formed hair, so the developmental process was not really affected by collagen 7 mutation. We again can see nice keratin 14 layer, we see nice keratin 10 layer, we could see human loricrene, we could see vimentin showing that there is dermal component, but we cannot really see collagen 7, which is exactly what we expect in these cell types. However, once we took our genetically corrected RDEB line, at least one of them, and again, just to emphasize, we tried them all, they all worked. So this in particular one. So you could see again, keratin 14, keratin 10, loricrene, vimentin, and now we see collagen 7. So just looking at organoids, we see that post-correction, we see the restoration of collagen 7 positive basement membrane. We also did electron microscopy of our organoids at this stage. So when I'm showing you just the corrected RDEB organoid versus uncorrected. We're looking at this area. Yes, sometimes it's very hard to interpret electron microscopy image, but here this is a basement membrane, and this is lumina densa where anchoring fibrils are. You see that in corrected RDEB organoid, you see this nice line. So it basically tells you that the basement membrane is fully organized. And now look at uncorrected RDEB. It's basically all over the place. There is really no 2 nice lines like we see in corrected organoids. And the control looks very similar to this one. So technically, again, it confirms that our genetic correction worked, and we see the restoration of basement membrane, at least in organoid. So importantly, we successfully isolated keratinocytes and fibroblasts from these organoids. So this image just shows you comparison studies here. This is fibroblasts and keratinocytes, primary fibroblasts and keratinocytes from skin biopsy of a healthy patient, stained for collagen 7. So you see both these cell types stained for collagen 7 because these cells express normally. These are primary cells from RDEB patients, keratinocyte and fibroblasts, and we don't really see much of a collagen 7 expression as expected. These are organoid-derived, genetically corrected RDEB keratinocytes and fibroblasts. Because they're genetically corrected, we could now see the restoration of collagen 7 expression in these cells. These images, flow cytometry images, just shows you that we have nice population of fibroblasts here that stain positive for CD90 and negative for CD104, which is the marker of keratinocytes. And these are our keratinocytes, negative for CD90, but they are positive for CD104. So technically now we have the right cell types. The question is, we still need to confirm their functionality to form human skin in vivo. Before we start testing these cells, we also need to think how we're going to deliver these cell types back to the patients. And there are a few skin transplantation techniques that we should consider. And the first choice was actually the generation of epidermal sheets from keratinocytes. So the reason why we initially focused on epidermal sheets is because there is a clinical trial being performed in Stanford, where also the researchers tried to correct RDEB. But instead of going through iPS cells, they just collect primary keratinocytes. They introduced a lentiviral vector carrying the full length of healthy collagen, like normal collagen 7. And again, just to note, lentiviruses can integrate whatever they want into the genome, so there is a risk of mutagenesis. Then they would grow these keratinocytes to a corrected keratinocytes, lentivirally corrected keratinocytes, to a sufficient number to produce these epidermal sheets that look like that. So the concern here is that it takes quite a lot of time to produce these epidermal sheets. They're more expensive to manufacture. They're very fragile. There is short shelf life for these epidermal sheets. So you have to transplant them within 2 weeks of their production. Otherwise, they go bad. And we deal with RDEB patients who sometimes get sick. They get infections. So we cannot transplant right away. So this will be wasted. We'll need to start all over again. That is difficult to transport. It's +4, and they have to be transported in the right condition. There is another issue I found out recently, is that in order to form good functional basement membrane, both keratinocytes and fibroblasts have to express collagen 7. So if we deliver just epidermal sheets with keratinocytes alone, we may not really achieve the best therapeutic outcomes for the patients because their fibroblasts will still be uncorrected. For the same reason, for that reason, we actually generated both genetically corrected keratinocytes and genetically corrected fibroblasts, so we can transplant them together. So if we again looking at the epidermal sheets, to introduce fibroblasts, we need to create another layer of the skin, the dermis. And it's possible, but it makes the production of these full thickness skin equivalents even more complex, more time consuming, more expensive, and they are more fragile, meaning that the whole transplantation approach will be very inconsistent. So for that reason, we actually entered into collaboration with Avita Medical. And Avita Medical developed a spray-on-skin device, which is now FDA approved for treating burn patients. So that's how this kind of kit looks like. You collect, like if you have a burn patient, you collect unaffected skin from this burn patient, dissociate it in a single cell suspension, put it into syringe, and apply to the wound. The cosmetic results from these transplantations are amazing. These patients have less inflammation, less infection, less scarring. So our idea now is, again, use our genetically corrected RDEB iPS-derived keratinocytes and fibroblasts and apply them to the wound of RDEB patients using this technique, using spray-on-skin technique. Another benefit for using this technique is we can freeze our keratinocytes and fibroblasts in a vial and keep them for as long as we need. We can easily transport these cells on a dry ice and apply to patients elsewhere. So in a way, we address the problem of limited shelf life of epidermal sheets, because technically we can produce as many of these genetically corrected cells from the patient in need of treatment, store it, and apply whenever it's needed, whenever the patient forms a new wound that we can use to apply these cells onto. So because we can now technically use cells in suspension, we can easily use our classical grafting chamber assay, to confirm the functionality of our genetically corrected iPS-derived keratinocytes and fibroblasts. So this is the outline of the procedure we use to confirm the functionality of our cells. We need genetically corrected RDEB iPS-derived keratinocytes and fibroblasts that we culture separately, which we already do. We can do all the necessary quality control tests. Then we combine these cells together in a cell suspension. After that, we use our silicone chambers, which we implant underneath of the skin of the nude mouse. So this is how the setup looks like. We cut a piece of skin on the mouse and insert the chamber into the skin. There is a hole underneath of this chamber like that. So the chamber technically sits on muscle fascia. Then on these top holes, you apply the suspension of your keratinocytes and fibroblasts. So you could even see a little bit of cell culture media right here. So think about the whole setup as a cell culture dish on a mouse, where mouse provides all the necessary nutrients. What happens then, very quickly, these cells self-assemble. Fibroblasts form the dermis and keratinocytes form epidermis. So we remove the chamber about 1 week after transplantation. That's how this wound area looks like. You could even see the formed graft inside, which we leave on top of the wound and let the whole area to heal until it looks like that. So after it heals about 6 to 8 weeks, we can cut off this piece, do sectioning, and see if we see human skin formed inside these graft. And we use immunofluorescent analysis for these purposes. So I'm just, for the sake of time, I'll just show you just one successful graft, but we technically now know that all of our cells now consistently engraft into the mouse, which was very exciting news for all of us. So this is how H&E looks like. Here on the left, you could see like the thin epidermis, while the rest is way thicker, because this thin epidermis is a mouse epidermis. Human epidermis is way thicker than the mouse one. But of course, we still need to do antibody staining that you see on this middle image. So we use mouse-specific keratin-1 antibody, and keratin-1 stains suprabasal layer of the skin. If everything is successful, keratin-1 will only stain the mouse tissue, but not the human. And you could see, like, again, I just cut the image, but you could see the green is right here because this is, again, mouse epidermis, which stops here because the rest is human epidermis. We also use human-specific loracrine, which stains higher level, like higher layer of the skin. And you could, again, see that it only starts staining right about here, but not here because this is mouse. But this is human. We know this is successful stratified epidermis that comes from human keratinocytes. We also see vimentin staining here, suggesting that our fibroblasts derived from organoids also engrafted. This antibody does not stain mouse dermis. So importantly, you could see that there are some weird structures in these grafts. The reason is this is one of the examples where we used fibroblasts very early after the isolation from the organoid. So, and this early grafting preserves dermopapilla cells, which are mesenchymal stem cells, which kind of sit underneath of the forming hair follicles. And they're very important for skin appendages to form. So in a way, our organoids can also provide the source of these mesenchymal stem cells in order to produce all normal skin appendages, including sebaceous glands and sweat glands, which are very important to to preserve skin metabolism and keep it healthy post-transplantation. So we also did electron microscopy image of this graft. And again, we see well-organized basement membrane and lumina densa. So again, suggesting that all our cells are functional, they produce functional basement membrane, and they can be used for transplantation in the humans once we adapt all of these technologies to the cGMP manufacturing with the help of Gates biomanufacturing facility. So in summary, we simultaneously reprogram and corrected the patient-derived RDEB fibroblasts using our patented technology. So keratinocytes and fibroblasts derived from these gene editing RDEB iPS cells can produce collagen 7 and reconstitute the full thickness skin, both in vitro. I'm not showing in vitro data, but in vivo is more striking, and in vivo. And we see these consistent results across our cell lines. Organoids can provide a more consistent source of high quality keratinocytes and fibroblasts for transplantation. So we're currently working with the GBF to adopt the production of modified mRNA iPS cells, genetically corrected iPS cells, and organoids to cGMP manufacturing. And importantly, because these technology, especially iPS and gene editing, are patented by our university, that can provide the platform technology for other researchers interested in using iPS cells to develop therapies in the future. And we know that there are few faculty on campus that work on developing iPS cell-based therapy for other conditions, which we think is very important and critical for the success of the Gates Institute. Also, our patented technological platforms are being used in stem cell biobank and disease modeling core that I co-direct with Dr. Kogut, my husband, as Dennis mentioned. So we basically provide the iPS reprogramming services, gene editing services to the research community on campus. So if you need any help with any of these cell types, we'll be happy to help. So at the end, I would like to acknowledge everyone involved in the project. So Igor and I basically have a joint lab